This comparative physiology presentation will be comparing the visual sensory systems of the cephalopoda and mammalia classes. Cephalopods, meaning headboards, showed up in the mollusk phyla during the Upper Cambrian period over 500 million years ago. The fossil record shows a broad diversity of these invertebrates over geological time, with only two subclasses of about 800 species inhabiting the oceans today. This is the Coleodea, which consists of squids, cuttlefish, octopus, and vampires, as well as the Nautilodea, which holds the only surviving cephalopods with external shells, the nautilus and the alonautilus. The class Mammalia appeared around 225 million years ago during the Triassic period, and is made up of about 4,000 species of vertebrates, categorized into 26 orders. Before comparing the cephalopod and mammalian eye, we first must understand what constitutes an eye. In its most basic form, an eye needs light-sensitive photoreceptor cells, comprised of light-absorbing pigment molecules, which form a cup-like shape. This cup-like shape is surrounded by pigment cells which aim the light to come into contact with the photoreceptor cell from one direction. The modern-day Coleodea have rid themselves of the slow bulky shells found on their primitive relatives, the nautiloids, reducing it to an internalized element. This freed them from the energy expenditure of producing and carrying around these external shells. They now rely on speed and visual manipulation to capture prey and avoid predation, which may be the reason for their complex size. Their vision is also used as a means of communication, which makes them the most vision-dependent group of all the mollusks. Before we look at the similarities and differences of the cephalopoda and mammalian eye, it should be noted that the eye types differentiate between the cephalopoda subclasses. The octopus, cuttlefish, and squid all have different eye morphology, with octopuses having slit-shaped rectangular pupils, cuttlefish having W-shaped pupils, and squids having round pupils. The living fossils, the nautiloids, have a much more primitive eye compared to the Coleodea. Their eyes are more like pinhole cameras, without a lens or cornea to focus light, and is in direct contact with the water, due to the lack of the cornea. So the major comparisons will be made between the Coleodea and the mammalian eye. Firstly, let's look at the morphological similarities and differences. Much like the mammalian eye, the cephalopod's eye consists of an iris, almost circular lens, vitreous cavity, and photoreceptor cells that produce the retina, which is where the Coleodea eye differentiates from the mammalian eye. Mammals have ciliary photoreceptor cells, where the membrane in the cilium is folded to increase surface area. Coleodea have rhabdomeric photoreceptor cells, common in most invertebrates. Within the rhabdomeric photoreceptor cells, the apical cell surface folds into microvilli to increase its surface area, and is pinnacle to allowing them to see polarized light. The microvilli are formed parallel to each other and contains the visual pigment rhodopsin, which assists in seeing polarized light due to the positioning of the microvilli. Keeping in mind, the majority of cephalopods are colorblind. There is also a large difference in how the Coleodea and the mammals focus their eyesight. Mammals use the muscles surrounding their eyes to modify the shape of their lens, whereas the Coleodea can control their lens to focus in or out. All cephalopods also have an inverted retina, opposed to a mammal's inverted retina. This means that the connection of the rhabdomeric photoreceptors and the optic nerve happens behind the retina in cephalopods. But for mammals, the optic nerve passes through the retina, resulting in a blind spot in mammals, but absent in cephalopods. A number of studies have discovered that the developmental stages of the vertebrate's eyes are influenced by 4 PAX6 splicing variants, affecting different genes, whereas insect eyes are governed by duplicating PAX6 genes. The same study was done on squids, finding 5 types of PAX6 splicing genes but no duplication, which was fluent in the insect. This is incredibly interesting, considering the cephalopods are a part of the protostome clade, which makes up many of the insect's phyla, including the cephalopods. This information leads to the understanding that the cephalopod and mammalian eyes are an example of convergent evolution. Convergent evolution can be between two organisms existing in separate geological locations and different evolutionary lineages, but both evolved similar adaptations. In this case, most mammals evolved in a terrestrial environment, while cephalopods were strictly marine. And although they have monumental differences between them, they both have evolved to have a similar eye. Thank you.